Hi everyone. My presentation is about role of TA plasmid. First, let's see about what is plasmid. A plasmid is a small extrachromosomal DNA molecule that can replicate independently of chromosomal DNA and is physically distinct from it. They are most often seen in bacteria as tiny circular double-stranded DNA molecule. They are extensively used in gene cloning experiment as vectors for the transformation of genes of interest into the host cell. Then let's see about role of TA plasmid. TA plasmid is also called as tumor-inducing plasmid. It carried by the bacteria Agrobacterium tumefaciens. This bacteria is a phytopathogen found in the soil. In the soil, it infects the plants and manifests in the form of crown gall disease. For the manifest and development of crown gall disease, TA plasmid is the important one. It is huge size plasmid ranging from 150 kb to 250 kb. The role of TA plasmid in biotechnology is that it may be converted into cloning vector by deleting the pathogenicity gene. The TA plasmid delivers a DNA fragment into the normal plant cells, causing them to convert into the tumor cell. Tumor cells also include genes for production of auxin and cytokinin hormones. Thus, their introduction into the plant aids in the production of plant's own nutrition machinery. Because of its comparatively high effectiveness during transformation, TA plasmid is the most widely employed in gene cloning. The tumor-inducing plasmid contains many components that aid in the efficient transfer of gene of interest into the plant cell. The DNA segment to be transferred into the plant is created by the tDNA sequence. tDNA sequence is the nothing but it is the region which is present in the plasmid. It is also called transfer DNA because this region is integrated into the plant genome. The tDNA carries the gene for tumor induction and therefore it is integrated inside the plant genome. It leads into the tumor formation. Genes in tDNA have plant regulatory sequence such as promoter, enhancer and other regions. The tDNA region must be transferred to the plant, which necessitates the presence of virulence genes. The tDNA region that has been edited to eliminate the genes that trigger crown gall development and replace them with the genes of interest. Apart from tDNA region, the TA plasmid consists of virulence genes that consist of 8 operons, 25 genes catalyzing the transfer of tDNA from agrobacterium inside the plant genome. The other region is ori site or origin of replication, which is required for replication of TA plasmid inside agrobacterium. The other genes for opine catabolism. Opines are nitrogen containing compounds required for growth and metabolism of agrobacterium. The tDNA region carries the genes for the tumor induction. These are right and left border of sequences which comprises of inverted repeats for the integration. Genes for auxin synthesis, it codes for enzyme monooxygenase and hydrolase that required for production and formation of auxin hormones. Genes for cytokine synthesis is a code for transferase that required for production and formation of cytokinin hormones. Genes for opine synthesis codes for synthase. All these three genes, including left and right border into the plant genome, are transcribed or translated into the respective enzyme and form plant hormones. When the plant cell produces these hormones in the exponential quantity, this drives the cell metabolism and cell division. The plant cell therefore divide uncontrollably, leading into the crown gall disease. The TA plasmid present in Agrobacterium tumefaciens, Agrobacterium rhizogenes, Agrobacterium ruby, and Agrobacterium vitis. For the bacteria to produce crown gall disease in plants, this TA plasmid must be present. Certain essential portion of TA plasmid including as the vir region which encodes for virulence genes and transfer DNA area aids in this process. Thank you.